All right, we have hit it, the last lecture. Today is Monday, December 7th. It's that weird combination of it feels like it's gone by so fast and it feels like we've been in the class forever. It's such a weird dichotomy. That seems to be what the semesters are like. Today, we have a ton of material to cover, so we're going to dive right in. We still have all of the autonomic pathways that we're going to talk about. The sympathetic ones, I guarantee one of those is going to be an essay question on the exam, and obviously the parasympathetic ones with things that we'll talk about as well. And then the good news is the rest of the lecture is really just kind of a review and adding a little bit more vocabulary to things that we already know. And then that's it. Wednesday is your lab and lecture exam 5. You know what to expect from that. Monday is finals week, so we don't have any lecture on that day, but I will have my normal office hours because on Wednesday I'll have all of my normal office hours during finals week, because on Wednesday we have our final, and again it's 100 multiple choice questions. You'll have 100 minutes to complete it, and again it must be completed during the class time. So you don't have to start right at 8 o'clock, but I encourage you to start sooner rather than later to make sure you give yourself enough time. And that way you can also deal with any technical issues as well. Again, at 12:35, it shuts down. So if you wait till 11:35 to start and then have 15 minutes of problems, then you're only getting 40 minutes to complete it. And so that's not a good thing. So make sure you are prepared for that. All right. Any questions?
You did say that even though you have an exam on Tuesday, you'll still be available, correct?
Yes, I will have all of my normal office hours. And again, obviously, after you've got your test on Monday, and then on Wednesday this week, and then my 431 has theirs on Thursday. So basically, I'm going to be spending the week between their lab and lecture exams and your finals grading furiously. So I will be doing that. So I will be around a lot. And again, like all of you, I'm quarantined at home. So I will be available potentially outside of that time. I'm not going to be on the computer 24/7, but I'll definitely hold my normal office hours. And depending on the time, I may be able to make other arrangements to meet with students if those times don't work for them. But yeah, I will be around. Final grades will be out once I get them completed. As I mentioned, so you guys have so you were complaining about an exam before the final. You guys have yours on Wednesday, and then you have a full week to the following Wednesday when your final is going to be. My 431 class, just the way the schedule and finals week fell out, they've got their lab and lecture exam on Thursday, and then on Tuesday, they have their final. So they don't have the full week to study the same way that you guys do. So I will be focusing on their exams first. But I expect by the end of next week, my goal would be by December 18th, maybe the 19th, that weekend. I hope to have them done. Typically, what I will do is I will post the grades onto Canvas. All of the grades, if you've done the science skills, if you've done the open lab, when I get those numbers from Jeff, the extra credit from that will be put in, the participation will be put in, the replacing your lowest lecture exam with the final, all of those things will be put in. I will do all the number crunching, post them all, and then I will leave them up for 2 or 3 days, giving everybody a chance to look at them so that if there's any questions or any concerns or anything that's missing or something along those lines, we can resolve those issues before I make them official. But my expectation is to make the grades official. My guess is probably before Christmas, depending on how things go, but definitely by the first of the year. Yeah, 10 weeks is a fast quarter system. Yep, Davis was nice that way. But then also, again, the pace is kind of brutal too. All right, any other questions? All right, let's get started because we've got a lot to cover. So we left off last class and we were taking a look at this octopus scary looking structure that was the autonomic nervous system map. And as we talked about, there are a couple characteristics about it that I think it does a really, really beautiful job of illustrating for us. the relationship between the two branches of the autonomic nervous system and their effectors, the two neuron pathways, the locations of their ganglia, and also their locations in the central nervous system. You'll see that while both of these innervate the eye and both of these innervate salivary glands, our preganglionic neurons are located in different parts of the central nervous system. And our postganglionic neurons in their ganglia are located at different locations as well. And that's one of the big differences between the sympathetic and the parasympathetic, is their locations of the cell bodies. Right? Where are their cell bodies located, both pre and postganglionic? And then what are the structures that are formed by the axons? Again, when we're talking about nervous system, we're talking about cell bodies, we're talking about axons. And so when we talk about these pathways, those are the things that we're going to be looking at. Where are the cell bodies located? What are the structures that are formed by the axons? And by doing that, we are going to be able to trace the paths to all of the different potential effectors. And we'll be able to do this in the sympathetic and the parasympathetic nervous system. Now, if you remember when we were talking about the anatomical differences between the sympathetic and the parasympathetic, remember we said that the sympathetic is very broad, very elaborate in its branching, forms lots of synapses, which again gives us our global path. So in our sympathetic, we'll be talking about general pathways because there's a lot of collateral ones to get there. But remember, we also talked about how the parasympathetic is very discrete, right, in its location. You don't typically cry and salivate at the same time. Right. So again, the nice thing about starting with the parasympathetic is it is much more straightforward. It is much more discrete, and the pathways are much more linear. So that is where we're going to start. Notice this pretty picture does a nice job of showing just the parasympathetic nervous system. And you can see how, again, remember the things that we talked about that are different are the location of the cell bodies and the location of the axons. In this case, as you can see, where's my annotations there? Come on, give me, there we go. When we look at the cell bodies for the preganglionic neurons, in our parasympathetic, you can see they're basically found in two locations. The first of those locations are in the brain stem. Now, of course, if we have cell bodies in the brain stem, those clusters of cell bodies, those clusters of gray matter, remember, are called nuclei. And these are the nuclei, as you can see from the illustration, whose axons form some of our cranial nerves. And as you can see from the illustration, those are cranial nerves 3. What was cranial nerve 3 again?
Ocular motor.
Excellent. Cranial nerve 7. What was that one again?
Vestibulocochlear.
Nope. 7
Facial.
was facial. 9 is which one?
Glossopharyngeal.
Glossopharyngeal. And 10, which is which one?
Vagus.
Vagus. Excellent. Notice also others of the second location is the lateral gray horn of the sacral spinal cord. So notice, based on the location of the preganglionic neurons, anatomically, the parasympathetic nervous system is known as the craniosacral branch of the autonomic nervous system, because its cell bodies of the preganglionic neurons are found in those two locations, either the brainstem forming cranial nerves or the lateral gray horn of the sacral spinal cord. So craniosacral. All right. Now notice we talked about how the axons coming out of the nuclei of the brainstem form cranial nerves. If you look here, you can see that the axons coming out of the lateral gray horn of the sacral spinal cord form what are known as pelvic nerves. Or they're also referred to, you'll see them in the literature, as the pelvic splanchnic nerves. P for parasympathetic, P for pelvic splanchnic. We'll see that splanchnic nerve in the sympathetic as well without the pelvic in it. So that's why I make a point of mentioning it there so that there's no confusion. All right, and that's it. Goal 1 is to figure out where the preganglionic neurons are and what their axons form. And now we know the 2 possible things that they can be, either part of one of our cranial nerves or part of that lateral gray horn forming those pelvic splanchnic nerves. All right. So we did that. The second thing we need to worry about, and I did this here. Okay, so let's do this here. The second thing we need to talk about is where the postganglionic neurons are located. Now, there are dozens of dozens of parasympathetic ganglia that contain our postganglionic neurons. However, remember, as we talked about, these are very close to or even inside of the effectors. So many of them, like for instance, the ones in the heart, are actually located in the wall of the heart or in the wall of the stomach. And if we've learned anything, it's that anatomists love to name everything. But even the anatomists kind of had to throw their hands up in the air on these. So for almost all parasympathetic ganglia, they just used a generic term for them. And that generic term is a terminal ganglia. So for most of our parasympathetic pathways, they just go through a terminal ganglia. There are only 4 of the dozens and dozens of parasympathetic ganglia. There are only 4 that actually get names. And all 4 of those are located in the head. And as you can see from the illustration, those are the pterygopalatine, the ciliary, the submandibular, and the otic. All right. Let's put these pieces together and write out some pathways. For this, I want to use our whiteboard. So what we need to do is we need to describe the pathways. Of course, we need to know where we are starting in the central nervous system. And of course, remember in the central nervous system, this is going to be where we find the preganglionic cell body. Then we need to know the structure that is formed by the axon of the preganglionic cell body. What is the structure that is formed by that? Need this down there, need this up here. Need that down there. We then need to know the name of the ganglion, which is, of course, where our postganglionic cell body is located. And then we need to know the effector. Now, obviously, we need to know also what the axon of the postganglionic forms. But here's the thing. For our postganglionic neuron, as we mentioned, that ganglion is located right at or inside of the organ. So in this case, basically for all of these, the axon of the postganglionic neuron just forms what we call a parasympathetic nerve. So really, all of these form a parasympathetic nerve. So there is no worry. So this is the true answer for all of them. So we'll just kind of put this here at the top and deal with that. All right, let's put the pieces together. As we know, there is option one of where in the central nervous system that we can be found, and that is in the brainstem. And as we know, in that brainstem, we can be the ganglia of one of four, or the nuclei, I should say, of one of four cranial nerves. So it could be the nuclei of cranial nerve 3. The nuclei of cranial nerve 7. The nuclei of cranial nerve 9. The nuclei of cranial nerve 10. And then also, we know it can be in the spinal cord, or more specifically, the sacral spinal cord. And of course, that's going to be in the lateral gray horn of our sacral spinal cord. Excellent. All right. So in our parasympathetic pathways, there's really only 5 different places you can start. The nuclei of cranial nerve 3, 7, 9, 10, or the lateral gray horn of the sacral spinal cord. And let's do the next part that is really easy. Guess what the axons of the cell bodies who are located in the nuclei of cranial nerve 3 form? Let me say that again. Guess what the axons of the cell bodies are in the nuclei of cranial nerve 3 form?
Oculomotor nerves.
Yeah, how about cranial nerve 3? Exactly. They form cranial nerve 3. Right? Guess what the axons of the nuclei of cranial nerve 7 form? These questions should be getting easier. There you go. Cranial nerve 7. Cranial nerve 9. And cranial nerve 10. And what did I say the axons of the cell bodies located in the lateral gray horn formed again? Parasympathetic, P for parasympathetic, P for pelvic. There we go. Pelvic, and remember, I also called them pelvic splanchnic nerves. You can just call them pelvic nerves if you want, but I do want you to be aware of this term splanchnic because it is going to come back to us. There you go. Just that simply. Now, remember, as I mentioned, most of the ganglia that are formed by the parasympathetic nervous system are terminal ganglia. But remember, I did say that there are 4 named parasympathetic ganglia. The first is the ciliary. The next is the pterygopalatine. The next is the submandibular. And the next is the otic. 4 cranial nerves, 4 named terminal ganglia. Perfect. Remember, for all of these are just going to form parasympathetic nerves coming out of them. So really, all we care about is the effector it is going to innervate. Anyone know what the ciliary ganglia happens to innervate? Term ciliary sound familiar to anyone? I'll get to that, Haley. We're working to it. It's part of a surprise. We're working on that. Anyone heard the eyeball? Exactly. So our effectors that are innervated by the ciliary ganglia is the smooth muscle of the eye. And which cranial nerve is responsible for that again?
3.
There you go. So if you wanted to dilate your pupil, that signal would start in the nuclei of cranial nerve 3, out the cranial nerve to the ciliary ganglia where it would synapse, then a parasympathetic nerve to the smooth muscle of the eye to cause it to constrict. Now, the pterygopalatine happens to be innervated by cranial nerve 7. Pterygo. What is pterygo? When I say pterygo, what does that sound an awful lot like? By the way, I brutalized Pterygopalatine. I didn't spell that wrong right at all. What is pterygo? When I say pterygo, what does it sound like to you? All right, what is that? You're absolutely pterygoid pilot. What is that? Yeah, what's it related to?
Pterodactyl.
Okay.
Sounds like pterodactyl.
It does. And what was special about the pterodactyl? Okay, let me rephrase that. What's the second thing that's special about pterodactyl? Because when I ask what the first thing that's special about a pterodactyl is, everybody says they fly. Yes, they flew. What was the other thing that was special about a pterodactyl?
It related to birds.
Well, remember, it looked like a bird because it had a huge, massive beak, right? And that's what, whether it's the pterygoid palate, whether it's the pterodactyl, it refers to the nose. So basically, the pterygopalatine ganglia innervates the mucous membranes of the nose. And it also innervates the lacrimal gland. What does the lacrimal gland do for us again? Tears, exactly. Now, as we talked about in the last class, we have 3 sets of salivary glands. And those 3 pairs of salivary glands are actually innervated by 2 different ganglia. Both the submandibular ganglia and the otic ganglia control different salivary glands. Now, you don't need to know the names of the 3 salivary glands, so you don't need to know which one is innervated by which. You just need to know that both submandibular and otic ganglia innervate our salivary glands. So then the question becomes, what innervates them? It turns out some of the neurons in the nuclei of cranial nerve 7, forming cranial nerve 7, go to the pterygopalatine ganglion, but some go to the submandibular. So cranial nerve 7 actually innervates both the pterygopalatine looking at it spelled wrong has been bugging the hell out of me. And some innervate cranial nerve 7. Cranial nerve 9, glossopharyngeal, innervates the otic ganglion. So there are the pathways involving our 4 named parasympathetic ganglia. So on the exam, if I asked you for the parasympathetic pathway that innervated the lacrimal gland, you should be able to describe that pathway, or one of the salivary glands, or the smooth muscle of the eye. You should be able to describe those pathways. Now, if you remember, we talked about how over 90% of our parasympathetic output travels out the cranial nerve 7, travels out cranial nerve 10, out the vagus nerve. And notice, wow, I did that really bad. I apparently have already taken the week off. All right, out the vagus nerve. The vast majority of our parasympathetic output goes out cranial nerve 10, and notice none of the ganglia for cranial nerve 10, for all of the potential effectors it communicates with, have a name. And remember, as we talked about in the previous part, when we were talking about the cranial nerves, we mentioned how cranial nerve 10 innervates most of the organs of the ventral body cavity. Well, now we can be more specific. The effectors that are communicated to by cranial nerve 10 include the heart, include the lungs in our thoracic cavity. It includes the stomach, the pancreas, the liver, gallbladder, spleen, the small intestine, and the proximal large intestine. So as we mentioned, the majority of the organs in the ventral body cavity are innervated by the vagus nerve. And any one of those pathways you would take, that vagus nerve communicates to a terminal ganglia, either right outside or actually inside of the organ, whether that organ is the liver or the spleen or the stomach or the small intestine, and et cetera, et cetera. Now notice we said almost all, but not all. Great question. Yes, if I asked you for the pathway to the salivary gland, there are two correct answers to that. So I could either ask you for both, or if I just asked for one, you could give me either. Sorry, I didn't see that question before. All right, questions on the vagus nerve. Well, notice we said most, but not all. So what is left? As we talked about, those splanchnic nerves synapse on a terminal ganglia from the lateral gray horn, and they innervate basically our reproductive organs, our urinary system, and our distal large intestine. So basically the rectum and the anus of the distal part of the digestive system. So notice if you want to pee, poo, or cum, it requires this pathway through the lateral gray horn, through those splanchnic nerves, those pelvic splanchnic nerves. Questions on that? And there you go. It really is just that simple. Those are all of the possible pathways for the parasympathetic nervous system. Very simple, very straightforward, very easy. And unfortunately, absolutely nothing like the sympathetic pathways, which are far more elaborate. All right, we've done it with words, which is an excellent way to do this. But again, as we talked about and look at the pretty pictures here, So again, here we see the 4 cranial nerves, 3, 7, 9, and 10, innervating our 4 named ganglia, pterygopalatine, ciliary, submandibular, and otic. And notice, as we talked about cranial nerve 7, some of its axons innervate the pterygopalatine, some innervate the submandibular. And as we talked about, all these are located in the head. Ciliary innervates the smooth muscle of the eye, pterygopalatine, mucous membrane, and lacrimal gland. Submandibular, notice, innervates 2 of our salivary glands, and the otic innervates the 3rd. But again, I don't care that you know that. So we talked about 90% of our parasympathetic output is the vagus nerve. And here we see how, again, it is truly that wanderer that goes to the heart, to the lungs, to the liver, to the gallbladder, to the stomach, to the pancreas, to the small intestine, to the proximal large intestine. And again, all of the ganglia associated with all of those organs don't have a name. They are just terminal ganglia. And like I said, some of those are actually inside the walls of the organ. And so they use the term intramural, right? Because if you think of intramural sports or sports within the walls of the school, intramural means within the walls. But again, you can just use the term terminal ganglia. I don't care. But we can't forget about the sacral portion of the cranial sacral parasympathetic nervous system. Again, it forms those pelvic splanchnic nerves that go to the rectum and anus, go to the urinary system, and go to the reproductive organs. And so again, you see these more discrete pathways to more discrete information. All right. Questions on that? Again, if this doesn't make sense, say something now, because like I said, the sympathetic is going to be a whole lot worse. All righty, excellent. See, look at that. In like half an hour, we whipped out the parasympathetic nervous system because it's simple and it is straightforward. What's not simple and straightforward is the sympathetic. All right, that's the ugly right side of this. So let's try to break it down in a similar fashion. The good news is, again, we need to worry about where our preganglionic neurons are located. And we need to worry about where our postganglionic cell bodies are located. And the one easy part about the sympathetic is where our preganglionic neuron cell bodies are located. All preganglionic sympathetic cell neurons are located in the lateral gray horn of the thoracic and lumbar spinal cord. In fact, specifically between T1 and L2, which is why it anatomically, our sympathetic is known as the thoracolumbar. Remember, our parasympathetic was called cranial sacral because that's where it came off the central nervous system. This one comes off the central nervous system just in the thoracic and lumbar region of the spinal cord. So it is the thoracolumbar. Now, we won't worry about what their axons form because, like I said, that's where things get a little bit wonky. So instead, let's talk about the postganglionic cell bodies. Obviously, they're all located in sympathetic ganglia. And there are 2 types. The first are what are called chain ganglia. You may also hear the term trunk ganglia. You may also hear the term parasympathetic paravertebral ganglia. These trunk ganglia, these chain ganglia, these paravertebral ganglia are paired, meaning that there's two sets, one on the left, one on the right. And they're to the sides of the vertebral column. That's where the term paravertebral. They basically run parallel to the vertebral column, like we see here on this illustration right next to it. They're paired ganglia, and there are many of them. The second type are what are known as collateral ganglia, or what are also known as prevertebral ganglia. These are singular, unpaired, and they sit right in front of the vertebral column. Notice on this illustration, they're pushed off to the side, but in reality, they sit in front of the spinal cord, which is why they're prevertebral, in front of the vertebral column. They're unpaired, and there's only 3 of them. And they are known as the celiac. Notice celiac, not ciliary. Ciliary was a parasympathetic. This one is celiac. Superior mesenteric, and inferior mesenteric. So we're getting a little bit of a closer look at our anatomy here. We've written this down. Let's continue to look at them more further. Again, anatomically, our sympathetic branch of the autonomic nervous system is known as the thoracolumbar branch. And again, remember, branches elaborately, widespread global effects. Here is a nice picture from your textbook that shows most of what we are trying to see here. Notice, as we talked about, there are 2 types of sympathetic ganglia. The first are these chain ganglia, and you can see now why they're called chain ganglia. They line up in a row in a chain connected to each other. As I mentioned, they're paired, so notice there's some over here on this side as well. And so they're run parallel to the vertebral column along the sides of the vertebral column, which is why they're also called paravertebral ganglia. This illustration doesn't show it, but will cheat. In that, notice there are axons coming here to the front. If we were able to see a little bit lower, down here, right in front of the vertebral column, would be where we would find our 3 collateral ganglia, or what are known as prevertebral ganglia, because they're located in front of the vertebral column. Oh, and I know a good place to see that. We'll take a look at that in a second after we talk about them. So, 2 types. The first are those chain ganglia or paravertebral ganglia, or you'll also see the term trunk ganglia. And as I mentioned, there are many of them. And since they run parallel to the vertebral column, not surprisingly, they are identified by the general region of the spinal cord. So we have cervical chain ganglia, thoracic chain ganglia, lumbar chain ganglia, sacral, and even a singular coccygeal. These are paired, meaning they're on the left and on the right. Here is a better illustration that shows that, where we see the ones on the left, we see the ones on the right. But there are a couple really important things we need to see about this. For starters, most people have 3 cervical chain ganglia. As you can see from the illustration, they are the superior, the middle, and the inferior cervical chain ganglia. So on an exam, if I had this illustration or a model like the one in the classroom that does an excellent job of showing this, and I had an arrow that pointed to this, you would need to say all of that. That is the superior cervical chain ganglia. This one is the middle cervical chain ganglia. This one is the inferior cervical chain ganglia. And while it is branching and elaborate and large global effects, if you look closely, for the most part, our superior cervical chain ganglia innervates everything for the head. So it innervates, notice the smooth muscle of the eye, salivary glands, lacrimal glands, etc. Basically anything smooth muscle or glandular in the head, it is innervated by the superior cervical chain ganglia. Whereas notice the middle and inferior innervate both the heart and the lungs, basically the organs above the diaphragm. So notice these 3 ganglia, and remember I said most people, because in a few people, the middle and inferior will actually fuse together into one ganglia. So there are a few people who just have 2 cervical chain ganglia. But basically just these 3 ganglia basically innervate all of the internal organs above the diaphragm. Let's take a look at that. We'll come down. Here we see another illustration that shows this. Again, superior cervical chain ganglia. If it is a gland, if it is a smooth muscle, erector pili, sweat gland, salivary gland, lacrimal gland, eye, innervates all the smooth muscle and all the glands of the head. Now, again, remember we talked about big global effects. So if you notice, there are some axons that come down this way. There is a tiny minor input into the heart, but it's not the primary way we innervate the heart. And again, that just shows that big global effect. So basically, if it is smooth muscle or glandular in the head, it is the superior cervical chain ganglia that innervates it. Notice if it is an organ in the thoracic cavity, basically above the diaphragm, primarily the heart and lungs, it is innervated by the middle and inferior cervical chain ganglia. And like I said, sometimes they fuse together. Those are the only 3 specific chain ganglia you will need to know. The rest you will just dump in a bucket we call chain ganglia. And the reason for that is everybody has a different number of chain ganglia. When we look at the rest of this chain, this down here, everybody has different numbers. Some people have 9 thoracic, some people have 12, some people have 10, some people have 4 lumbar or 5, some people have 4 or 5 sacral. So on an exam, if I were to point at one like this, do I know whether that's thoracic 10 or lumbar 2? No, there's no way to know that. So all the rest of these we just call collectively chain ganglia. With one exception. Notice everybody just has one coccygeal. And remember that coccygeal actually connects to the other one. If we go back a couple pictures, let's go back a couple pictures and we look closely at this. Notice right down here, while for the most part, these ganglia are parallel to each other, notice the 2 coccygeal ganglia come together. And when they come together, they form a structure known as the ganglion impar. So these 2, let me clear my drawing, put a box around it. These 2 coccygeal ganglia come together to form a structure we call the ganglion impar. Now, regardless of how many thoracic, lumbar, sacral, and coccygeal chain ganglia a person has, they all do the exact same thing. All the rest of these chain ganglia innervate all of the structures associated with the skin. Notice that means sweat glands, the sebaceous glands, sudoriferous glands, both epicrine and merocrine, eccrine, smooth muscle of the erector pili, smooth muscle of the blood vessels. It even innervates the adipose, releasing or storing more adipose. Remember, we talked about releasing that energy, right? That glucose to help us to deal with that stress response. And if you think about it, they don't just go to random parts of the body. Remember, we learned about the dermatomes. The dermatomes are a specific region of the body innervated by a specific nerve. Well, these are the branches that go to that specific region to control the sweat glands just in this band, or the erector pili in just this band. They are precisely arranged using our spinal nerve to get to those destinations. All right. So notice we've done all the organs above the diaphragm, all of the superficial surfaces of our skin. What's left? What haven't we gone to?
Organs below the diaphragm.
Exactly. What we haven't done is gone to the organs below the diaphragm. And that is what our paravertebral or collateral ganglia do. Here's another excellent view of those chain ganglia. Notice the chain ganglia are right here paired to the sides of the vertebral column. And notice one other thing that we've talked about as well. Notice there are 2 rami that go to it. Remember that gray ramus and that white ramus. Those rami communicantes are the pathways to and from those chain ganglia. Here's another view showing those chain ganglia. And again, the gray and white ramus feeding into them along the side of our spinal cord. All right. What we're left with then is our 3 collateral ganglia: celiac, superior mesenteric, and inferior mesenteric. Now, I always find it ironic. What is the superior most prevertebral ganglia? Is it the superior mesenteric that has superior in its name? Would
No.
I be asking the question if it was? No. The celiac is the superior most. So the celiac is actually more superior than the one called superior. So why is the one called superior? Why are any of these given the names that they're given? You should be able to answer this, Haley. Why do these names sound familiar?
Is it their length?
Not a bad guess. Nope. Those 3 names don't sound familiar to you? Aren't you taking 431? So why do those 3 names sound familiar? Wow, I didn't mean to put you on the spot. I really have, haven't I?
How about arteries?
Blood vessels, exactly. Absolutely. These are blood vessels. The nerves and blood vessels tend to travel similar pathways. And so the celiac ganglia sits right next to the celiac artery or the celiac trunk. Superior mesenteric ganglion sits right next to the superior mesenteric artery. Inferior mesenteric ganglion sits next to the inferior mesenteric trunk, or artery. So all of these things are named by the blood vessels they are sitting next to. You're absolutely correct. So that's why they have these weird combination of names where the superior one isn't the superior one. It's the superiormost, celiac is, but they're named by the blood vessels they're sitting next to. Absolutely correct. Now, these innervate very specific, hold on, where'd my list go? Bring that back. These innervate very specific organs. So again, remember, there's big global effects, but in general, the celiac innervates the liver and gallbladder, the stomach, the pancreas, and the spleen. All of the proximal organs in the digestive system. The superior mesenteric innervates all of the small intestine and the proximal large intestine. And the inferior mesenteric innervates the reproductive organs, the urinary system, and the rectum and anus. So if you think about it, we have 6 specific named sympathetic ganglia that you're responsible for. Superior cervical chain, middle cervical chain, inferior cervical chain, right? Celiac, superior mesenteric, inferior mesenteric. 3 of those are chain ganglia, and 3 of those are collateral ganglia. And then the rest are just the rest of the chain ganglia. And now we know what all of those, so I guess technically 7, because the 7th would be a generic chain ganglia. And the good news is we know what all of those 7 ganglia innervate. So that's going to be our goal. How are we on time? We are good. Okay, excellent. Our goal is going to be to put these pieces together. I'll get to the, here we go. Let's start here. Because we did that, we talked about all of that. Excellent. Here is an illustration. Now, again, if we were actually in the actual classroom, I would actually have you draw this. But again, I'm not going to force you to use the whiteboard to draw these things, and I'm not going to evaluate your grading on the whiteboard. But I will still draw it out because I still think it's a good way to visualize the pathways. Hopefully, this is not the first time you've looked at this. Hopefully, you heeded my advice and looked at this over the weekend. And your textbook does a really nice job of describing actually all 4 pathways. However, your textbook really only emphasizes 3 of them. There are indeed 4. There are 4 precise sympathetic pathways that I'm going to teach you that are basically going to be the 4 pathways you get to any effector of the body. And I guarantee you that one of these 4 pathways will be an essay question on the exam. So what we will do is something similar to what we did for the parasympathetic. We will see where the preganglionic neuron is. We will see the structures that its axon forms. We then know its synapses on a ganglion, where there is a postganglionic neuron. We have to identify that ganglion, identify the structure made by its axons, and determine what effector it innervates. So we will do the same thing that we did before and do that. And actually, we'll take a couple minutes to do this first. Let's go and get to our starting point. Our starting point for this is going to be the anatomy. So let's actually draw the anatomy first. I need you out of my way. I need you out of my way. We start with our spinal cord. We know coming out of the spinal cord, we have our dorsal root with its dorsal root ganglion and the ventral root. Now, again, remember this is a motor pathway, so we're not going to be using that dorsal root at all, but it doesn't hurt to draw it in there. We know we then have our spinal nerve. And then we know our spinal nerve branches, not 1, not 2, not 3, but 4 times. Now, of course, we know we need to start in our lateral gray horn. We know this here is our ventral root. We know this is our spinal nerve. What are the name of the 4 branches that come off the spinal nerve?
Dorsal ramus, ventral ramus, then the white, and then gray.
Excellent. Dorsal ramus, ventral ramus. White and gray. And again, which one is which? Which one's closer to the spinal cord?
Gray is closer.
Excellent. Excellent. Now we know that these are going to connect to a chain ganglia. We don't worry about identifying which one it is yet. And we do know they are part of a chain, so we do have to cheat a little bit here. And again, this is where my artistic skills will fail us miserably. but for argument's sake, let's pretend that this is, oops, the ascending chain going down to another chain ganglion there, and we'll make this the ascending chain going up to another chain ganglion there. So I know this should be in a line, but again, I'm a horrible artist, so we'll have to deal with that. However, we know that there is another type of ganglion too. Sitting in front of the spinal cord up here in the front is our collateral ganglion. And we know that these need to connect. Maybe we don't know that yet, but we're going to learn it. So this is our collateral ganglia, one of three. Oops. I need to put you here so that I can put that there. There we go. We have now named most of the structures here that are here, not all of them, but most. And so, of course, the other piece that we need is ultimately we need to know which effectors we are going to be communicating with. So we can describe an effector, describe the pathway. And as we know, there are going to be 4 total pathways. Oops. That we are going to be responsible for, and we will identify them, we will describe them, and I will draw them for the heck of it. All righty. This is going to be a lot of information, but this is going to be our starting point. Sure, that's with that. That's with that. You can put this up there. Put that there. Put that there. Your brain hasn't exploded yet, but it's about to. But you've got the right idea. Because we are all about to explode our brains, I think the best thing for us to do before we dive into this, again, it's a little scary, but it is pretty straightforward. But we definitely need a break. We need a chance to decompress, and I need some coffee. So let us go ahead and take our first break now. Looks like it is 9:08. So before we dive in, we'll take our first break. We'll take a 15-minute break. That is going to be, what is that, 9:23 then? We will restart, and I will start the recordings at that time. All right. Any questions before we take our break? This is college. You're adults. You don't have to be here. All right, excellent. So, yes, more coffee for Laura, more coffee for me. Actually, my first cup. I don't like to start the day with it, but first break is when I get my coffee. And so let's do that and meet you back here in 15 minutes. I'll see you guys in 15 minutes. All right. So as I mentioned, there are going to be 4 pathways that we are going to be responsible for on this exam. You need to be able to identify the neurons, and those are easy, preganglionic, postganglionic. You know they're multipolar. We also need to talk about the neurotransmitter they're going to release. Now, the good news is, remember, the one basic thing that is true for all of these is where our preganglionic neuron is located. So when we talk about our preganglionic neuron, and let's use blue for that and make it smaller so it fits, and we'll sneak it in up here. When we talk about our preganglionic neuron, for all of the pathways, where is that preganglionic neuron going to be found?
Lateral gray horn.
Yeah. And if you think about it, there are basically three things we know about it. We know it's multipolar. All right. What is the condition of its axon? Is it myelinated or unmyelinated?
Myelinated.
Myelinated. And remind me again what neurotransmitter it releases?
Acetylcholine.
Excellent. Or let's say releases acetylcholine. So that is true for all of our pathways, and all of this is true for our pathways. So all four pathways, the nice thing is they're all going to start in the exact same way. We start with a multipolar neuron whose cell body is located in the lateral gray horn. And when its axon leaves, which way does it go out, the ventral root or the dorsal root?
Ventral.
Ventral, right? Because we know the ventral root is motor. And because I didn't write dorsal root and I did write ventral root, that's probably a good safe bet that that's the way it is going to go. Ventral root, of course, comes to the spinal nerve. All right. So the beginning of this is going to be true for all of our pathways. So that is a good starting point. One of the easiest ways to think of in terms of our pathways, the four possible pathways, is to think of in terms of what effector we are going to. The first effector that I want to go to is something associated with the skin. Remember, this means all the glands of the skin, the erector pili muscles of the skin, the blood vessels associated with the skin, all of those types of things. So any of those, if I gave them to you, a sudoriferous gland, a sebaceous gland, an erector pili muscle, if any of those were the potential effectors, then this is going to be the pathway that we're going to take to get here. Now, based on this, with a skin effector, do you remember which of our two types of ganglia we want to synapse in? In a chain ganglia or in a collateral ganglia?
Chain.
Chain.
Chain. Excellent. So, if you think about it, our myelinated axon of our preganglionic neuron has two choices to get to that chain ganglia. It can go down the gray ramus or it can go down the white ramus. And being that it is myelinated, which one of those two structures do you think that myelinated, covered with fat axon is going to form?
White.
The white ramus, exactly. So from the spinal nerve, it travels down the white ramus to our chain ganglia, where it can then synapse. Now, it certainly can synapse on the very first chain that it comes to, but remember, it has the ability to ascend before it synapses, or it can descend before it synapses. And in reality, it probably does all three, because again, big, huge global effects. But whether it ascends, whether it descends, or whether it synapses right away, we know it is going to release acetylcholine. And when it releases acetylcholine, is that acetylcholine going to be positive or negative? Excitatory or inhibitory?
Excitatory.
Excitatory. There you go. Perfect. So it's going to release acetylcholine. That acetylcholine is going to be excitatory, and it is going to stimulate our postganglionic neuron. Now, of course, let's do some writing here. Our postganglionic neuron. And again, this is going to be true for all of them. They are going to be found in some type of ganglion. different ganglion, but in a ganglion. They are also multipolar. But what do we know about their axons?
Non-myelinated.
Unmyelinated or non-myelinated would be fine. And what neurotransmitter do most release?
Acetylcholine.
Do the postganglionic sympathetic nerves?
Norepi.
Norepinephrine. Excellent. Norepinephrine. I'm just abbreviating these for simplicity here. Obviously, you'll write these out on the exam. So, our preganglionic neuron synapses on this postganglionic neuron, releases acetylcholine, stimulates it, and then it needs to send a signal to the skin to tell the erector pili muscle to stand up, or to tell that sweat gland to sweat. In a specific strip of the body, one of the dermatomes. So if this axon needs to get back to the skin, that means it's going to need to go out either the dorsal ramus or the ventral ramus. Right? Coming out this ventral ramus would get us out, for instance, to the finger. This would take us to the hairs on the small of our back to be able to stimulate those. So, is there a structure that our unmyelinated axon of this postganglionic neuron could use to get back to those dorsal and ventral rami so that it could get to the skin? Yeah. So this axon is going to go out the gray ramus to either the dorsal ramus or the ventral ramus until it gets to that effector of the skin. And once it gets to the skin, it's going to release norepinephrine. And again, whether it's excitatory or inhibitory is going to depend on the effector. If it's the erector pili muscle, it's excitatory, making the hair stand up on end. Right? If it's the blood vessel, it's excitatory, constricting it, drawing the blood away from the skin. Right? And so on and so forth. So if you think of all those physiological effects, it would either excite it or inhibit it depending on the situation. So notice our first pathway involves synapsing on the chain ganglia, and then out the gray ramus to the dorsal or ventral ramus. And just that simply, and not too scarily at all, we have described the first pathway. We've described it, and we've drawn it, seen the structures. Now, again, you don't have to draw this on the exam. If you want to, you're welcome to. But you would just need to describe the pathway. Preganglionic neuron, whose cell body is located in the lateral gray horn. Its axon forms the ventral root, the spinal nerve, the white ramus, where it goes to the chain ganglia and synapses, releasing acetylcholine and is excitatory. It synapses on the postganglionic neuron in that chain ganglia, whose axon leaves the gray ramus to the dorsal or the ventral ramus to go to the surface of the skin. All right. Just that simply, we have done pathway 1. Questions on that? All right, let's talk about pathway 2. Pathway 2 is to an organ, internal organ, above the diaphragm. Now, if we're going to an internal organ above the diaphragm, where are we going to synapse here? in a chain ganglion or a collateral ganglion?
Still chain?
Yeah. We're still going to synapse in a chain ganglion. But notice here, it's going to be one of three. It's either going to be the superior cervical, the middle cervical, or the inferior. And we can be even more specific than that. If we synapse in the superior cervical chain ganglion, what are the possible effectors that we would be innervating? I would be one. What else?
Smooth muscle in the head?
Yeah, smooth muscle in the head, lacrimal glands, mucous membranes of the nose. Don't put heart on this one. We know there's a minor for the heart, but let's ignore that minor for the heart. But you absolutely have the right idea. All smooth muscle and glands of the head. So note if I said lacrimal gland, if I said your mucous membrane, if I said the eye, all of these pathways specifically are going to be the superior cervical chain ganglion. Whereas if it's the middle and the inferior cervical chain ganglia, what was the possible effectors there?
Heart and lung.
Heart and lungs. So notice we still need to synapse in a chain ganglia. So really, the beginning of this pathway for pathway 1 and pathway 2 are identical. Notice for both of these pathways, 1 and 2, hold on, I'm going to use red for that, green. For both 1 and 2 are preganglionic neuron is identical. So that makes this really easy. We start with a preganglionic neuron whose cell body is located in the lateral gray horn. Its myelinated axon leaves the ventral root, forms the spinal nerve, forms the white ramus, where it goes to a chain ganglion, where it can either ascend or descend to find the specific chain ganglion we want. And let's say, for argument's sake, we're going to the lacrimal gland. If we were going to the lacrimal gland, which specific chain ganglion would this be?
Superior.
Cervical, superior cervical. Don't forget to say cervical. It would be the superior cervical chain ganglion. Excellent. It would synapse on our postganglionic neuron in that superior cervical chain ganglia. It would release acetylcholine, which would be excitatory. What's different in this second pathway is where our axon of this postganglionic neuron goes. Instead of going out the instead of going out the gray ramus, it leaves out a structure to get to the internal organs above the diaphragm. Out a structure called the sympathetic nerve. So instead of forming the gray ramus, it is now forming a sympathetic nerve where it goes to an internal organ above the diaphragm. And again, depending on what it is, It could be excitatory or inhibitory. If it goes to the heart, what does it do to the heart? What is that norepinephrine? Remember, we're sympathetic. Norepinephrine does what to the heart?
Slow it down.
Because when that bear with an axe comes in the room, I want my heart to beat slower.
Isn't that epinephrine that speeds it up?
Epinephrine and norepinephrine, remember, chemically are identical and functionally are identical. This is our sympathetic pathway. This is our fight or flight, right? This is going to make the heart beat stronger, faster. However, if it goes to the smooth muscle of the lungs, what does it do to the smooth muscle of the lungs? Does it excite it so that the muscles constrict it, or does it inhibit it so that the muscles dilate and we get more air in? It relaxes it, inhibits it, absolutely. Right, again, that's why I carry my EpiPen with my daughter everywhere. Make her heart beat faster, make her airways dilate, so hopefully she lives. So there you go. Just that simply, we have our second pathway. Notice the only difference between the first pathway and the second pathway is where this postganglionic axon goes. If it has to get to the skin, it goes out the gray ramus to the dorsal and ventral ramus. If it has to get to an internal organ above the diaphragm, it goes out a sympathetic nerve. Oh, hold on. Wonder how far back I can do this undo. Cool. I didn't save the first picture. So now I've saved the first picture. Look, it's like a movie. There we go. All right. Perfect. Questions on that? Hey, I'll try not to take that personally. All right, excellent. So let's go to, no offense taken. Come on. Erase, 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 erase. All righty. Pathway 3. Pathway 3 is going to be to an internal organ below the diaphragm. Now, if we want to go to an organ below the diaphragm, where do we want to synapse that in that case?
In the collateral ganglion.
Excellent, in the collateral ganglion. And how many collateral ganglia are there?
3.
3. What are they?
Celiac, superior mesenteric, and inferior mesenteric.
Excellent. So, if I'm going to an organ below the diaphragm, an internal organ below the diaphragm, I have to synapse in one of these 3 collateral ganglia. Now, which organs does the celiac ganglion innervate again?
Liver, gallbladder, pancreas, or spleen.
and stomach.
Yep, there you go. Bladder, pancreas, spleen, and stomach. Excellent. Superior mesenteric innervates what?
Small and most of the large intestine.
Intestine and proximal large intestine, which means the inferior mesenteric innervates the distal large intestine. And again, by that, we pretty much mean the rectum and the anus. But what else does it do? Oh, there you go. Reproductive system and urinary system. Excellent. Perfect. So let's say the essay question was to innervate, let's say, the small intestine. We know our preganglionic neuron starts the same way, with a multipolar cell body in the lateral gray horn of the spinal cord. Its myelinated axon comes out the ventral root, forms the spinal nerve. And notice there's really only one place it can go, and that is the white ramus. However, this time we don't want to synapse in the chain ganglia. So instead, what happens is it keeps right on through, passing through the chain ganglia without synapsing. It, of course, is still myelinated all this way. And when it leaves, it forms a structure. This myelinated preganglionic axon forms a structure that is known as the splanchnic nerve. Remember, pelvic splanchnic was in the parasympathetic. Here, splanchnic is in the sympathetic. So P for pelvic splanchnic, S for splanchnic. That's how I remember the two of them apart. This is going to travel all the way to the collateral ganglion, where, of course, it is going to synapse. And what neurotransmitter does it release again?
Acetylcholine.
Acetylcholine. And is that acetylcholine excitatory or inhibitory?
Right.
Excitatory, exactly. And it does that on our postganglionic neuron. As we know, the postganglionic neuron's multipolar. Its axon is unmyelinated. And when this unmyelinated axon leaves, it just forms a structure we call a sympathetic nerve. Notice these unmyelinated postganglionic axons, whether they are above the diaphragm or below the diaphragm, are just called sympathetic nerves. Now, I think I said we were going to the small intestine, right? So not only does it synapse in a collateral ganglion, but which specific collateral ganglion would it be?
Superior mesenteric.
Excellent. I like that. Superior mesenteric ganglion. I like that abbreviation. Actually, no, I'm not going to do that. Superior, because I want it to be here. Mesenteric ganglion. Excellent. So specifically on the superior mesenteric ganglion, and then, of course, it would innervate our squiggly small intestines. What neurotransmitter is it going to release? Norepinephrine. And since we're here at the small intestine, what do you think? Is our sympathetic excitatory or inhibitory to our small intestine?
Inhibit.
Yeah, it's going to inhibit it, absolutely. But in parentheses, again, could be excitatory depending on the effector. So in this particular case, it would be inhibitory because we're specifically going to the small intestine. But again, it always depends on what the effector is. And there you go. Just that easily, we have done our third pathway. Questions on that? See, these aren't as scary as you guys first thought, huh? Okay, maybe. All right, excellent. But now comes the important question. And that important question is, what the hell is left? We've gone to all surfaces of the skin. We have gone to all the organs above the diaphragm. We've gone to organs below the diaphragm. What could possibly be left? Well, but see again, do that. Like for instance, the legs, all of the skin and the blood vessels of the muscles, those are all part of that first pathway to the skin. So that first pathway to the skin would take you to an erector pili muscle or to the blood vessels of the legs. So it wouldn't be that. Getting closer with a gland, but one gland in particular. What gland? There we go. The one gland that is special to the sympathetic nervous system and gets its very own special pathway is the adrenal gland. Now, of course, remember where the adrenal gland sits. It sits on top of the kidney. So it is also sometimes referred to as the suprarenal gland. Both of those are acceptable terms for that adrenal gland. What makes this pathway special is where the synapse occurs. Now, for this, let's cheat. I need to do some quick erasing. Erase. Oops, hold on, I didn't want to erase that. No. All right, I'm confused. All right, hold on. I want to get rid of that. I want to get rid of that, that, and that. So here is the kidney. Oops, nope. And sitting on top of the kidney is the adrenal gland. And clearly that's not drawn to scale. All right. Now, like many organs we have seen in the body, the adrenal gland has a chewy nougat center and a candy-coated outer shell. When we have that type of organization, what do we call the outer region again?
Cortex.
Cortex, excellent. And what do we call the inner region? Medulla. And I didn't leave myself enough room to label that as the medulla, so we'll just do it that way. With the adrenal gland, it really doesn't truly have its own ganglia. Instead, our preganglionic neuron actually synapses in the cortex of the adrenal gland. So in this case, our postganglionic neuron is actually located here inside the cortex of the adrenal gland. If you think about it, this pathway is almost more similar to what we see in the parasympathetic, where the ganglion is actually inside the organ or next to the organ. That's kind of what we have here. So this is actually kind of similar to what we see in the parasympathetic. So the way we get there, oops, one more thing I need to erase, that right there. This pathway starts the same way as 3. We have a preganglionic neuron whose cell body is located in the lateral gray horn. Its myelinated axon forms the ventral root, the spinal nerve, the white ramus, to a chain ganglia, where it passes through the chain ganglia. And again, hold on, not the superior cervical, but it passes through a chain ganglia, out the splanchnic nerve, specifically to, in this case, the celiac ganglion, where its myelinated axon passes through the celiac ganglion all the way to the cortex of the adrenal gland, where it finally synapses. Of course, as we know, it releases acetylcholine, and that acetylcholine is excitatory. On our postganglionic neuron, which is located in here. Oops, I'm an idiot. Not in the cortex, I'm sorry. Synapses in the medulla. All right, get rid of that. Oh no, not that, just that, perfect. Ugh. There we go. I just want to get rid of that. Perfect. All right. Now I can cheat and redraw my medulla. I apologize for those of you following at home, playing the home version of this game. Sometimes when I'm talking and I'm thinking about what I'm saying next, I'm not thinking about what I'm actually saying the first time. So let's fix this. There we go. Bigger medulla. There we go. Synapse is in the medulla, where it releases acetylcholine that is excitatory. On our multipolar preganglionic neuron, and this multipolar multipolar postganglionic neuron then releases epinephrine and norepinephrine, and probably about 80% epinephrine and 20% norepinephrine into the blood. And when it releases that epinephrine and norepinephrine into the blood, where does it go?
To the kidney?
Okay, that's one place that it goes. But once
It goes
a
everywhere.
chemical, once a chemical like this gets into the blood, it goes everywhere. Remember, part of the goal of the adrenaline in that adrenal gland is to give us that big global stress response. Once this gets into the blood, it affects your whole body all at once. And it sustains it. Like we talked about, when there's that big loud bang of that car backfiring, you instantly know right away that that was just a car backfiring, but your heart rate doesn't instantly go back to rest, right? Your nasal cavity is suddenly very clear, right? Because that adrenaline is released and you get that big, huge global response. So this fourth pathway, a separate pathway to the adrenal gland, releases that adrenaline into our blood, giving us that big global response. And that is our fourth pathway. Questions on that? Well, as long as it makes sense to you, Haley, that's the only thing that matters. And I have actually saved these pictures. I will post these pictures for the basic anatomy and then the 4 pathways so that you have that to look at as well. Oh, that's very nice. I like that. All right. That's it. We can look at the lecture and see this now and go through it. Notice here's the pretty pictures from your textbook to kind of show these things. But really, all we're going to do now that we've gone through it, because it's so much information, is keep beating this dead horse over the head until we fully understand and get it and are comfortable with it. So notice, again, when we think about our sympathetic pathways, we can think of them in terms of where they synapse. Notice for pathway 1, this synapse is in the sympathetic chain ganglion. All right, we have a preganglionic neuron whose cell body is located in the lateral gray horn. Myelinated axon leaves out the ventral root to the spinal nerve and then goes down and forms our white ramus, that lateral communicante. There, it synapses in the chain ganglia. Remember, it can ascend or descend first before it synapses. Then that postganglionic unmyelinated axon leaves out the gray ramus, going to the dorsal and the ventral ramus. And from there, remind me again, what are the effectors of this first pathway?
Skin?
Yeah, basically anything. So again, smooth muscle and glands of the skin. So here we do in words what we did with pictures in our first pathway. And again, you can furiously write these down, but you should have this on your notes and on your lecture handouts. So again, don't feel too stressed to write it all down. Because again, there's nothing new here. This is what we just did. Pathway 1. All right, questions on that? All right, remember, however, pathway 1 is not the only one that synapses in those chain ganglia. Pathway 2 also synapses in our chain ganglia. And it starts the same way. Preganglionic neuron in the lateral gray horn. Myelinated axon leaves ventral root, forms the white ramus. Synapses in the chain can travel up and down. Notice this is identical. The only thing that's different is where our postganglionic axon goes. For pathway 2, instead of going up the gray ramus, it goes out that sympathetic nerve. And if it goes out the sympathetic nerve, what are the effectors? And Haley's already got it. In this case, the effectors are all the internal organs above the diaphragm. Perfect. So again, no new information. We've already covered this. And really, the only difference between pathway 1 and pathway 2 is what are postganglionic unmyelinated axon forms. Now, we've seen it with words. Here we see it with the pretty pictures. This one is not from your textbook, but I like it because it shows things on both sides. Now, again, it reminds us that this is paired. Notice it shows pathway 1 here on the right. Synapse in the chain ganglia, out the the gray ramus to the nerves. And over here on the left, it shows pathway 2, synapsing and out the sympathetic nerve. Does that mean that all of the skin goes out the right and all of the internal organs go out to the left? No, they're just showing the 2 different pathways on the 2 different sides in this. But I like this because we see the paired chain ganglia. We see the single unpaired collateral ganglia. And notice it even shows us the adrenal gland that we're going to use on this. So this picture is not from your textbook, but it's a really nice picture that I like it. So that's why I make it here so it's available for you. Here's the one from your textbook. Notice here it shows, again, going to the skin. And then they even have a picture where it shows it ascending first and then going to the skin. So it shows that first pathway, both ascending first or just synapsing right away. Pathway 3, remember, is going to synapse in a collateral ganglia. It starts the same way. Lateral gray horn, ventral root, spinal nerve, white ramus. But remember here, the difference is that it does not synapse within the chain ganglia. Instead, it exits, forming that splanchnic nerve. And it goes to our collateral ganglia where it synapses. From there, sympathetic nerves leave and innervate the organs below the diaphragm. Stomach, small intestine, bladder, so on and so forth. Again, I like this picture that's not from your textbook because it shows how nerves from both sides go to that single unpaired collateral ganglia and then leave. I like the picture from your textbook too, but your textbook bugs me. Notice here, lateral gray horn, ventral root, spinal nerve, white ramus through the chain to a ganglion. However, notice, it even mentions the splanchnic nerve. Notice what they've done here. Here, they say such as the celiac. Now, granted, they are saying such as. They're not saying this is the celiac, but they're saying such as the celiac. And look at what organ they're innervating. The intestine. Again, for example, intestine. So again, they're saying such as, they're saying an example. But does the celiac innervate either of your small or your large intestines? No, it would have been nice if they'd paired up the right ganglion with the right organ. But again, they did say such as, they did say for example, but again, it would have been nice if they'd paired them together. But you got the idea. Splanchnic nerve, synapse in a collateral ganglion, and then out that sympathetic nerve to that effector below the diaphragm. And lastly, our adrenal gland, the suprarenal medulla, lateral gray horn, ventral root, white ramus, do not synapse in the chain ganglia, travel through the celiac ganglion, but do not synapse there. Go to the adrenal gland, where there you synapse in the medulla, and you release those hormones into the blood. And again, we've done it with words. Here it is with the pretty picture. This is the pathway. Your book talks about this pathway, but it doesn't draw it the same way. So that's why I make a point of emphasizing. Your book does a really nice job of emphasizing the first three pathways, but it doesn't necessarily specify this one. Now notice, now that we come back to this scary looking octopus, we can really see and appreciate what a nice job it does of showing these processes. Notice again, starting in the lateral gray horn, we would go out the white ramus, we would go out the ventral root to the spinal nerve, to the white ramus, to a chain ganglia, where we could synapse right away, or we could ascend or descend. Notice if we wanted to go to the skin, we would synapse right away, or ascend and descend, then go out that gray ramus to the dorsal and ventral ramus to the skin. Or we could synapse in the superior cervical, where we go to the organs of the head. Or we could synapse in the middle and inferior cervical chain, where we go to the organs above the diaphragm. Or We could pass through the chain without synapsing to form splanchnic nerves, to go to the celiac, to innervate the liver, the gallbladder, the stomach, the spleen, the pancreas. We could pass through our chain ganglia to the superior mesenteric to go to the small intestine or the large intestine. Or we could go out the inferior mesenteric to the reproductive organs, the bladder, the kidney. But notice one more thing. We can also start in the lateral gray horn, pass through the chain ganglion without synapsing, go to the celiac ganglion and not synapse, continuing as a preganglionic axon all the way to the adrenal medulla, where we then release into the blood. Notice this scary looking chart does an amazing job of actually showing us all 4 pathways. Now that we know what to look for, we even see that preganglionic neuron going through the celiac ganglia to get to the adrenal gland. Again, there's lots of ways to think about this. I've just put all this information in here to help you to synthesize it. Again, when you think of when you come out of our preganglionic neurons, what you can do, you can synapse right away. You can ascend or descend and then synapse. You can pass through it to the sympathetic nerve, the ganglia, or you can pass through it and go all the way to the adrenal gland. Or you can think of it in terms of, again, where are the cell bodies located? Preganglionic neurons are all in the spinal cord, T1 through L2. So where's our preganglionic? Either in the chain ganglia, either in a collateral ganglia, or in the medulla. If you're in a chain ganglia, you're either going to the organs above the diaphragm or the skin. Collateral ganglia goes to the organs below the diaphragm. And if you synapse in the adrenal medulla, basically you're releasing hormones into the circulation that affect everything. All right. Lastly, we can think of it as what are innervated spinal nerves from the gray ramus go to the dermatomes. Sympathetic nerves from the chain ganglia go to the organs above the diaphragm. Splanchnic nerves go below the diaphragm and to the adrenal gland. So again, all these slides have no new information on them. They're just different ways of organizing the information to help you find the best way so that it makes sense to you to how to use this information to understand these pathways. And I guarantee there's going to be a question on the exam. Describe the pathway to effector X for the sympathetic nervous system. Identify all the structures, identify the neurons, characteristics of the neurons, neurotransmitters they release, and their effect. All that thing we talked about is going to be here. All right. And there you go. Even the simple one, notice the pathway to the adrenal gland goes all the way to the adrenal gland without synapsing. Here we have collateral ganglia. Here we have chain ganglia. Here we have terminal ganglia. These graphs, while they're scary when you first look at them, once you understand this information, they actually make a tremendous amount of sense. All right, for the most part, this is really most of the new information. What we really need to do with the remaining stuff is kind of synthesize information we've already talked about. We're going to add a little bit more vocabulary to the things that we've already learned. And there are going to be a couple new things that we'll talk about, some of it which you may have already had. But the heavy lifting for the day is done. That is the good news. So the good news is we are done with the heavy lifting. But we do still have a little bit left to finish this up. But again, I think we definitely need to decompress after that, give you some time to relax and synthesize. So let's go ahead and take another break. We will restart at 10:30. And at 10:30, we will start the recording again. All right. So any questions on that before we take our break? All right. See you guys in 15 minutes. So, oh, that was the other thing I wanted to show you. Hold on. Should have set this up during the break, but as always, your other resources, and there's plenty of these. But the Cosumnes River Visual Anatomy Lab is useful. Oops, where did you guys go? There you are. So here we see the chart that is the nervous system. And again, when we first look at this, this is a little bit scary. But if you look closely, you can see a lot of the stuff that we have been talking about. So for instance, right up here, let's start with the easy things to find. Notice here you see the chain ganglion. You can actually see the chain ganglion going down. You can't quite see the ganglion in par, although this chart does show it. And then they come back up the other side. So you can see that there. And so notice here, here, and here are those three cervical chain ganglia that we've talked about. The other thing that I want to show you from here is that if you look closely, you can see right here, and right here are the three collateral ganglia. And again, I appreciate that this is small, the charts are not nicer, but the point that I want to remind you of is that, again, the chain ganglia are also the collateral ganglia, and then the prevertebral, by the chain ganglia are also the paravertebral along the side parallel, and the collateral ganglia are prevertebral in front of it. There is also in here, this does a nice job of showing the plexes that we talked about for the spinal nerves. Oh, here's the one I want to show you. This is the other one, the other chart, which again, you can see here is the inferior, middle, and superior cervical chain ganglia. Here we see the rest of the chain going down to the bottom. You don't see the ganglion in par where it comes together, but this also shows the caudae equina very nicely and some other things on it as well. So there are some good resources that do a nice job of showing you some of the anatomical characteristics that we are responsible for. So these sympathetic and parasympathetic nerves that we've talked about, after they leave and are going to the effectors, form big elaborate networks. And as we know, a big elaborate network of nerves is called a plexus. So there are a handful of plexuses that are formed. Again, often named from the blood vessels that they are associated with. And some examples of these include the cardiac, the celiac, the inferior mesenteric, hypogastric. You don't have to write those down. I'm not going to hold you responsible for those. But there is one that I do want to talk about. Notice here, this illustration does a nice job of showing most of the chain. It shows our collateral ganglia where they're located here. But the thing that I want to show you on this particular illustration is right here, where the celiac ganglion and the superior mesenteric ganglion, let's make that bigger. Where the superior mesenteric and celiac ganglia are located. Collectively, this is a structure that is known as the celiac plexus. You may not have heard the term celiac plexus before, but you probably are aware of it maybe by its other name. It is also sometimes referred to as the solar plexus. Or it's also your fifth chakra. Again, maybe you didn't know the name of it, but maybe you are aware of its function. Because not only do sympathetic and parasympathetic nerves form the elaborate plexus that is the celiac plexus, but your friend and mine, the phrenic nerve, coming off of the cervical plexus that comes down and innervates the diaphragm, is also forming part of this plexus. So maybe you were sitting around on the quad one day during gym in elementary school, talking with someone of the other sex, thinking you're doing pretty well, when suddenly someone kicks a soccer ball, and without you being aware of it, it hits you in the stomach unexpectedly. And especially if it not just hits you in the stomach, but right under the ribs, what ends up happening to you as a result of that?
Knock the wind out of you.
Yeah, you get the wind knocked out of you. Suddenly you have gasping for air, right? Bent over, doubled over in pain. And as a result of that, feeling like you're going to die. Now, do you die from that? No. But what happens is that pressure in that location right under the ribs basically disrupts this celiac plexus, and it disrupts the function. So yes, that means your stomach is functioning a little less efficiently and your liver is functioning a little bit less efficiently for a short period of time, which you're not aware of. But it also means your diaphragm stops functioning. Now, luckily, you have accessory muscles, so you are able to elevate and depress your ribs to be able to get air into and out of your lungs, but it sure as heck feels like you're going to die. And that's from that disruption of the activity of that celiac plexus. So if you haven't had the wind knocked out of you, you probably know somebody who has. And now you know why that has occurred. It's because that pressure wave disrupted the activity of this celiac plexus. So that's the only one we really need to be aware of. And here, again, when we remove the pesky person, we see, again, this nice plexus, this elaborate network formed by the chain ganglia, formed by the sympathetic and parasympathetic nerves, and other things like that. But like I said, the celiac plexus is the only one that you need to know the function of. All right, let's talk about some more new things that are not new. As we know, neurons release neurotransmitters at a target cell. And most of those target cells are other neurons. For these to communicate, our first neuron has to release a neurotransmitter. That's way too big now. And our postsynaptic cell has to have some type of chemical receptor. All right. If, for instance, our neuron releases acetylcholine, then we know that our target cell has to have some type of acetylcholine receptor. Oops. On its cell body. Certainly not a new or, odd piece of information. We were fully aware of this. And in fact, if you think about it, for acetylcholine, we even know that there are two types of receptors. Because remember, as we've talked about, if, for instance, this target cell, let's do this. If this target cell, for instance, was skeletal muscle, what kind of effect does that acetylcholine have again?
Excitatory.
Excitatory. Whereas if this target cell is in said cardiac muscle, then what kind of effect would it have?
Inhibitory.
So we know some neurons release acetylcholine. We know some cells have acetylcholine receptors. No new information there. And we know that there are two main types of acetylcholine receptors because we can get two different effects. Again, no new information. So what we're going to add is some new vocabulary. A neuron that releases acetylcholine is called a cholinergic neuron. And all cholinergic neuron means is a neuron that releases acetylcholine. Yeah, let me make that bigger. A neuron that releases acetylcholine is called a cholinergic neuron. Therefore, a receptor that acetylcholine binds to is a cholinergic receptor. Again, no new information, just a couple of new labels, really a new vocabulary term. Something associated with acetylcholine is called cholinergic. Now, the last little bit, as we know, there are two types of cholinergic receptors. It would have been really nice if they named them A and B, because A and B would be a really easy way to tell them apart. But we do, yeah, exactly, Anonymous hates you. We do need a way to tell them apart, and here's the problem. If I release acetylcholine and acetylcholine binds to it here, we know that acetylcholine is going to bind to it. If I release acetylcholine here, I know acetylcholine is to bind to it. So if I have some brand new receptor that I don't know what it is, so let's say I've got a new cell, and this new cell has a new receptor that I don't know what it is. If I release acetylcholine and acetylcholine binds to it, do I know which of these two receptors it is? Can I tell whether it's A or B? True, it could be that it's excitatory or inhibitory. And as it turns out, this one is always excitatory. But here's the problem. This one here on our cardiac muscle is sometimes inhibitory, and it's sometimes excitatory. So just the fact that it's excitatory or inhibitory isn't going to be enough to tell us which is which. Now, these are chemoreceptors. And as we learned in the skeletal muscle system, things like tetanus can bind and activate a receptor. There are toxins that can also bind. And as it turns out, different toxins bind to both of these receptors. This first receptor that we called receptor A, a special toxin called nicotine can bind to this receptor. Where might nicotine come from?
Just gonna take a random guess and say cigarettes.
There you go, or tobacco. You absolutely have the right idea. Nicotine primarily comes from tobacco. And it binds this first receptor, not the second one. So because of that, that receptor A is named by the toxin that binds to it. They call it a nicotinic receptor. And it's named after the toxin that binds to it. And as I mentioned, it is always excitatory. Receptor 2, there is a poisonous mushroom toxin called muscarine that will bind to this receptor and again can slow the heart down or have other effects like that. And so receptor type 2 or B is a muscarinic. And as I mentioned, it's a little bit more complicated in its effects. Sometimes it can be excitatory, and sometimes it will be inhibitory. So cholinergic neurons communicate with cholinergic receptors, and one of two types, nicotinic or muscarinic. Let's do it again. Cholinergic neurons are neurons that release acetylcholine. If you think of the neurons that we've talked about that release acetylcholine, remember we said all preganglionic neurons in both sympathetic and parasympathetic. all parasympathetic postganglionic neurons. Some of our sympathetic, remember, particularly ones associated with the skin, like to the sweat glands, like to our blood vessels, like to our somatic motor neurons. So if you think about it, there are really 4 types of cholinergic neurons that we've talked about. Our somatic motor neurons, all of our autonomic preganglionic neurons, all of our parasympathetic postganglionic neurons, and then some of our sympathetic. As we talked about, 2 different types of receptors. Nicotinic, where it's nicotine, always excitatory. These are the ones, for instance, that are found on our postganglionic neuron, are found in our neuromuscular junction. Remember, anytime we said where a neuron releases acetylcholine and we said it's always excitatory? Like at our skeletal muscle, like on those postganglionic neurons, anytime it's always excitatory, it's always a nicotinic receptor. Muscarinic receptors are stimulated by that mushroom toxin, muscarine, and it can be excitatory or inhibitory. And these are the ones that are found on the parasympathetic pathway and our sympathetic sweat glands. Remember, I told you the story about my former student whose boyfriend likes to eat wild mushrooms, and occasionally he gets some poisonous ones. Those poison is most likely muscarine. So again, remember what we talked about, what happens. It stimulates our parasympathetic nervous system. So his pupils constrict, his heart rate goes down, his airways constrict, and notice also he gets really sweaty as a result of that. So he gets really sweaty, his airways are closing down, his heart rate's slowing down, his pupils are constricting, all from eating that poisonous mushroom. Because it binds and stimulates these muscarinic receptors. Think of nicotine instead. Think of that first time you had a cigarette, right? You stole one out of grandpa's backpack, took it out into the woods. You were 7, right? Lit that baby up. Right. So, okay, one of the things that could have happened is you could have gotten nauseous as a result of it. Because again, it suppresses our digestive system. That can upset your digestive system that way. It can make you very jittery, right? Because it stimulates your neuromuscular junctions. I'm just kidding. No, I wasn't 7. It was 6.5. I lived in Ohio. There wasn't anything else to do. All right. We were encouraged. No, I'm just kidding. But the point is, but remember also, it can be addicting. It can stimulate that insula that we talked about, causing an increase in neurotransmitter release there, which is what leads to the addictive nature of nicotine as a result of it. It causes dopamine to be released, so you feel calm and relaxed as a result of it. You're more alert and things along those lines. So there's all those, and again, if you think about, we think of nicotine as being a bad thing. Nicotine in and of itself as a chemical isn't a bad thing. Right? It is all the other things you have to ingest to get it. The smoke, all of the toxins, everything that is in those cigarettes. And especially now with today's tobacco, it's been basically super hybridized to make it incredibly rich in nicotine, to make it incredibly addictive, so that you're doing all these other harmful things, inhaling all these dust particles and other toxins and things along those lines, right? So that those cigarette companies can make gajillions of dollars off of you. So don't smoke. I'm not encouraging smoking, just saying that nicotine has these effects. All right. Of course, then neurons that release acetylcholine are cholinergic. So a neuron that releases norepinephrine is adrenergic. And if you think about it, there's only one adrenergic neuron that we've talked about. Most of our sympathetic postganglionic neurons release norepinephrine. So that's easy. When it comes to receptors, there are 2 main classes of receptors. And again, it would have been nice if they had named them A and B. And it turned out they did. Of course, again, an anatomist can't do nice things. So they didn't quite call them A and B. Instead, they called them alpha and beta. But alpha and beta is still A and B. And as it turns out, there are two specific types of alpha receptors and three specific types of beta receptors. And I know you probably haven't thought of this before, but I know you know this. Probably UC Davis has got a fair amount of anatomists. They're all horrible, horrible people. Yes, I agree. So let's talk about these type of adrenergic receptors. Starting first with the alpha. Remember I mentioned there are two alphas. The first specific one is alpha 1. It is always excitatory, and it is by far the most common type. So again, if you think of our effectors, most of our smooth muscle of our blood vessels have these alpha receptors. Our digestive system has these alpha receptors, right? All these are areas where we have these alpha receptors. Conversely, alpha 2s are always inhibitory, decreasing the metabolic activity. So for instance, these are the ones that are found in the salivary glands. So when you release norepinephrine and epinephrine, right, your mouth gets dry. Your nasal cavity dries up, right? The blood vessels to your skin constrict so that the blood goes to other parts of your body. But there are also the betas, and remember there are three types of betas. The first are the beta 1s. These increase metabolic activity. They're always excitatory, and they're almost all found on the heart. Now, you may not have ever thought of any of these things in any terms whatsoever, but one of the things that you are aware of is when you're carrying your 3 bags of groceries up of 2 flights of stairs to get to your apartment, your heart has to beat faster and stronger to get the blood to your body so you can carry those 3 bags of groceries up to the 2 flights of stairs. And that is absolutely positively a good thing. Unless you just had heart surgery. If you had just had heart surgery, do we necessarily want our heart beating stronger and faster every time we get up and move around? No. So after heart surgery, what are most people given in the forms of medication?
Beta blockers.
Beta blockers. And guess what beta blockers block?
Beta receptors.
Beta 1 receptors, absolutely. So that now when you're walking up the stairs, your body releases adrenaline, your body releases norepinephrine, but it can't bind to those heart cells. And so your heart rate stays low, your heart strength of the contraction stays low, and we don't put that extra stress on the heart.
With question, would the beta blocker can you block specific beta receptors, or will it block all the beta receptors?
Just beta 1s, right? Beta 2s are found in a different location, and beta blockers don't block the beta 2s. Beta 2 relaxes smooth muscle and specifically is in the respiratory system. These 2 beta receptors is why I walk around with that EpiPen anywhere my daughter goes. Again, if she comes in contact with a nut, if she goes into anaphylactic shock, I need to jab her with that EpiPen so that her heart beats stronger and faster, so that the smooth muscle of her airways dilate so she can get the oxygen to her body, she can get that oxygen and that blood to her brain, and she can survive. And that's the beauty of those beta blockers. The beta blockers just block the beta 1s, right? So it's not like you have trouble breathing when you take that beta blocker. It just stops your heart from beating faster and stronger. Super, super important. However, probably of all of the receptors in the body, the one that is currently being studied the most, that is having the most money thrown at it, are our beta 3s. Beta 3s are found primarily on adipose tissue. And what your beta 3s do is they stimulate lipid breakdown. They help to encourage the cells to release adipose, or release triglycerides, the fats, and help in the metabolism of that fat. It also increases your overall body temperature. So you are using your fat, breaking down your triglycerides, increasing your metabolism, and making it more likely that you metabolize fats. Why bother with all of that pesky exercise when you can just take a pill that will literally melt your fat away? All right. Many scientists believe that these are the miracle pill that will make them incredibly rich. If we can find a way to stimulate these beta 3 receptors, then you can just melt your fat away. Sure, it'll increase your metabolism. You'll sweat all over the place, but you'll look beautiful. And that's really all that matters. Because as we know, the only thing that matters in life is looking beautiful. Oh, I'm sure there's loopholes. I'm sure there's all sorts of loopholes. But like I said, who cares what's going on the inside of your body as long as you're beautiful on the outside? All right. But are there people spending, or companies, I should say, spending gajillions of dollars to study these to try to find out? I know the anatomists just are realists. They understand how the world works, right? All you have to do is get onto TikTok or get onto OnlyFans to see how shallow the world is, right? You don't need an anatomist to tell you that. Anatomists just take advantage of it. All right. But so again, a very, very interesting field of study that they're looking at here. And again, I know this is a lot of vocabulary and it seems just like vocabulary for vocabulary's sake, but we've already kind of hinted at why this is important. Like we talked about, once we understand how something like this works, we can use it to our advantage. All right? We can manipulate it. And as we've learned, we can manipulate these types of receptors one of two ways. We can find a chemical that binds and blocks it, or we can find a chemical that binds and activates it. We talked about this with tetanus. Well, not with tetanus. We talked about, with tetanus and with curare, right? And curare, we talked about tetanus. We talked about curare. We talked about these things and the effect that they would have. Oh, and your book's got a nice table that goes through all of these receptors. But the reason this is important is because once we understand where these receptors are located, we can use them to your advantage. A receptor agonist is a chemical, and again, by chemical, I mostly mean toxin. Because again, it's not something that is normally produced by the body, but it is a toxin. And again, but many of these toxins are medications or things like that that we use, but basically it is a chemical that binds and activates a receptor, basically mimicking the effect of the neurotransmitter. When we talked about muscles, we talked about tetanus. That's what tetanus did. Tetanus bound and activated it, making that muscle contract. A great example of this is phenylephrine. Phenylephrine is an alpha-1 agonist. This is what's in cold medicines. It constricts the blood vessels going to your mucous membrane. If you have less blood going to your mucous membrane, you produce less mucus. And if you produce less mucus, your nose stops running, the congestion of your nasal cavity goes down, and you can breathe a little bit easier. All right, that's basically how cold medicines work. By stimulating this receptor to constrict those blood vessels to stop mucus production. We talked about nicotine, right? Increases dopamine, releases epinephrine, makes you feel good, right? Makes you alert. These are things that bind and activate it. But remember, these chemicals can also bind and block. And if they bind and block, we call them a receptor antagonist. This is putting the gum in the keyhole, right, to inhibit the neurotransmitter effect. Classic example most of you have experienced is atropine. Atropine blocks the muscarinic. Remember, this is the one that causes your pupils to constrict, right? Because hopefully everybody in here has had an eye exam. And as you know, eye exams are done by optometrists. And as you also know, optometrists are incredibly horrible, lazy people, right? For starters, they only have their appointments at noon on sunny days. And then when they're looking in your eyeball, they're too lazy to tilt their head a little bit to the left or tilt their head a little bit to the right. So instead, what they do is they put drops in your eyes. And those drops in your eyes, like atropine, block the parasympathetic input to your eyeball. As we know, most organs have autonomic tone, sympathetic and parasympathetic coming in. But if we block the parasympathetic, then just our sympathetic to our iris comes in and your pupil dilates dramatically. And then when they shine that blinding bright light into your eye, can your poor little iris constrict to block that light? No. And that gives them allegedly a really good view of your retina. But I say that if they just tilt it a little bit to the left or right, they should be able to see it fine anyway. They're just lazy. Of course, like I said, they only do these appointments at noon on sunny days. So then they kick you out of the office after that and you're blinded by the sunlight. They give you those crappy little wraparound plastic sunglasses that don't do anything. Like I said, they're almost as bad as anatomists. Horrible, horrible people those optometrists are. I have nothing against podiatrists. No, look, it's just well, I am an anatomist. That's the funny part of that joke. And I just like making fun of optometrists because they're incredibly lazy people. I once had a optometrist sit me in her chair and she spent 4 hours doing every single eye procedure she knew on me. For 4 hours, I sat in that chair. Yeah, and they $1,000 for a pair of glasses, absolutely. So like I said, horrible, horrible people. All right. So again, that depresses the parasympathetic nervous system, dilates the eye, also decreases mucus production and all those things. And of course, we talked about the beta blockers, those heart medicines that reduce the effect of norepinephrine. So there you go. And again, your book's got a nice chart that talks about receptor agonists and receptor antagonists as well. And that is everything you need to know about anatomy and physiology for 430. That is true. Optometry school is very, very expensive. But yes, that is everything you need to know for anatomy and physiology. For 430, you still have 431 to go. So you're not done with anatomy and physiology, but you're done with 430. All right. Any questions? I missed it. I'll have to look back at the video. All right. So, as I mentioned, guys, that's all I have for you. I want to thank you for an amazing semester. Definitely a unique semester. This has definitely been a unique experience. I hope we've gotten some of it out of it. I hope to see you guys back again for 431. Yes, that's a good point. We have an exam on Wednesday. We have the final on the following Wednesday. After that, if you were taking me for 430, I want you to go ahead and take a good 45 minutes, maybe even an hour and a half off. And then I want you straight studying the cardiovascular system, because if you're taking me for 431, we are starting with the heart. So again, good hour and a half. Tell you what, 2-hour break. Take a full 2-hour break and then start right working on the through break on the cardiovascular system on the heart. All right. All right, guys. We don't have any more classes, but I will have my office hours. I'm going to be really, I'm always good at responding to emails, but especially try to be responsive over the next week and so that if you have any questions, I can help you to be successful. Because again, while the lecture is done, you guys have a tremendous amount of work still to do. I appreciate your time and effort in this, and I wish you great success with me or wherever you go into 431 from this. Enjoy your break, and from there, I wish you guys great success. So take care. I will see you hopefully in office hours or email me. Otherwise, have a good break. Have a safe break. Be responsible. Be smart. Listen to the doctors. And I'll see you guys in the new year. All right. Bye.
Thank you.